Professor, can you please share with me the general link that went for the public? Because a couple of other people emailed me for the link just now. I only have the co-host link.
Yeah, I'm trying. Let me see. I'm giving you through chat. Okay.
Okay, sure. That helps.
That is coming as a registration link.
Oh,
Yeah. Sarah, do you know? Sarah is here. How to create a link for?
so usually once they got registration, they would get the emails, right?
Yeah. Yeah. Okay. I think we can start as 10:01. Welcome everyone for our first webinar. This is a webinar series for sustainable management practices, practical insights for healthy soil, funded through FFAR grant. And we have our first speaker, Dr. Franz Lubers. Just want to let you know that another speaker cannot join today because of some medical emergency. So Alan is going to talk about almost 35 to 40 minutes. So I'm just going to introduce him. Alan, can you in meantime share your screen? So Dr. Alan Franz Lubers is a research ecologist with the USDA Agriculture Research Service on the campus of North Carolina State University in Raleigh, NC. He has more than 30 years of research experience, starting with a master's program in Nebraska, PhD in Texas, and postdoctoral position in Alberta, Canada, and as a soil scientist in Georgia, North Carolina. During the past several years, he has conducted much of his research on private farms to better understand the practical evidence for soil health management. Alan, the floor is yours.
Thank you, Asmita. Welcome, everyone. Thanks for joining this webinar. It's a pleasure to be here to be a part of this project that is funded by the Foundation for Food and Agriculture Research, and the funding going to the Carbon Farming Alliance for Research and Management at Ohio State University. I get some specific funding as part of this project from Cotton Incorporated, and I really appreciate their dedication to my research over the past decade or so. So with that, I'm going to continue and really talk about the soil buzzing. Well, what does the soil buzzing mean? The soil is full of life if we really stop and take the time to understand it. This image here is a simple image that has actually some components of the life that we would desire in soil. It has a green plant in there. That green plant is going to have roots in the soil. It's going to then grow and it's going to die. And there are plant residues in this photo here. Those plant residues are important. They are important because they feed the microorganisms, because most of the microorganisms in soil are heterotrophic, meaning that they get their food from the plant material or plant or animal residues that are in soil or on soil. And so they are that food source is necessary. You know, you've heard the slogan that if you build it, they will come. Well, you have to build the plant material on the soil. And so by incorporating that material, it decomposes very rapidly, and then it leads to very little of the food source left for them. So this image here and this accumulation of surface material here, along with some fungal material, is really the embodiment of soil health, in that we want the soil to always have a steady supply of food source so that they can thrive and they can continue to grow. There is another thing that's here in this image that we often don't really appreciate, and that's that hand there, a person that is actually oftentimes that leads to soil degradation, unfortunately, because of the way we manage our land, and that we allow it to erode away, or we let nutrients either increase or decrease at an unsustainable level. But that hand can also be good for the soil, and that's where we have to embrace this in the future, is that our management, however that management occurs, is going to need to be good for the soil. So the human dimension is really important there. Oftentimes, soil health is teamed rightly with earthworms, and earthworms in the soil can be really quite the ecosystem engineers, and they are important because they form the soil, they mold the soil, they create aggregates that are resistant to erosion, they transform the plant material into usable nutrients that the plants can use. And so those earthworms can be a really good sign, but they don't really have to be there, but they are when they are there, it is a good sign. The other part of it is that really the important part is the part that we don't really see. Those of the bacteria, the fungi, the actinomycetes, those organisms in soil that are so small we need a microscope to see them. But they live off of the plant material. They're on the living plant material as well. But when the plants die, they are decomposed, and they can decompose fast or slowly. But that decomposition is really a fundamental basis of soil health. That's that transformation. And this image here shows a lot of earthworm casts on the surface of the soil. So the earthworms were present in this soil, but there are also the bacteria and fungi that are transforming those nutrients and those carbon compounds. So when we talk about soil health, really the focus is on the function. We want soil to produce plants and food. That's the fundamental nature of agriculture. And we've been able to do that very effectively over a long period of time historically. But we also want soil to be able to supply water, nutrients, and plant growth promoting compounds. And that's an important function that if we keep a focus on that, then we'll understand that soil is really providing something for humans. We need soil to be physically stable so that we can build on it, or that we can traffic over it so that we can produce our plants. And that's important. Otherwise, it might wash away and it won't have that stability. We want soil to be able to cycle nutrients, to be able to take plant material that has grown, that decomposes, and that feeds the biology, and then the new plants that are coming afterwards. We want soil to also protect water quality. When we think about it, we allow water to pass through soil, which we oftentimes call dirt. And that soil actually cleans water, and it improves water quality by cleaning the water that goes to the groundwater. So soil that as a medium is really important. We also want soil to be able to store carbon and nitrogen. Those are the two elements that are of fundamental importance for agriculture, because the carbon is really the largest concentration of elements in the organic matter, and nitrogen is an important part of soil fertility, and it's also a relatively large component of organic matter. So storing that carbon and nitrogen in soil is a good thing. With that carbon and nitrogen in the soil, it actually provides a habitat for organisms. Now, those organisms can be small, from the bacteria, to very large. They can be moles, but there are a lot of in-between things, of beetles, of spiders, of earthworms, and various other collembola and other critters that are just in the soil. And it's great to take the time to actually sit down and lie down on the earth and actually see what's in the soil. So we also consider soil to be a reservoir of biodiversity, and that's an important function that we don't really appreciate it, and we need more metrics to actually understand, well, how much biodiversity is really being provided by soil. There's a filtering of elements so that the soil can be cleansed, and actually that the toxic elements don't accumulate. So these are functions that the soil, in interaction with the organic matter and the microorganisms, really have a large role to play to actually transform the soil into something that is actually very good for us. And so these functions in totality are important. but there are also some that are just really, really important. And I guess that's what my focus that I wanted to in my research in the past decade was really about how do we produce plants for food, and how do soil cycle nutrients. And so those are the primary ones that agriculture farmers and others in agriculture really are focused on. But there are a lot of other functions that we could consider. And this test that I'm going to describe a little bit more detail for you is called soil test biological activity. It's got this name because we want to understand what is the biological activity, but we need to also standardize it into a soil test so that it's consistent, so that we don't just get any random number, but we get a consistent number that means something. That biological activity could really potentially influence all these other functions, but the focus of the research that I've done is really on that production and the cycling of nutrients. And we've made some attempts at some of the other functions, but there's a whole lot more research that really could be taking place on that front. So this soil test biological activity is a standardized test. It really is characterizing the organic resources needed for soil microorganisms to be active. And so ground that is covered is really, really important. We need to think that ground cover is protection for the soil. It's also creating a habitat for the organisms. And that exhaling of CO2, the carbon dioxide coming from soil, which we can measure, and that is natural and it will happen, but it depends on how rapidly it will happen. Because if we plow the soil, it will happen very rapidly in a big burst. But now, theoretically, there could be considered this buzz, and that's the buzz that I'm talking about. It's not necessarily a physical buzz that you can actually feel, but it's something that is conceptual, that there's the activity in the soil, and we need to actually understand how that is occurring. So I'm going to explain a little bit about the soil test biological activity. We basically measure it from this graphic here, where we have the cumulative carbon mineralization, and we use milligrams per kilogram of soil as our units, and we incubate the soil for a certain amount of time. And this graphic here shows soil from 0 to 4-inch depth, 4 to 8-inch depth, and 8 to 12-inch depth. So the test is really about when we take a soil sample, we dry it, intentionally dry it out of the field, and then we re-wet it, and we create what happens during that re-wetting is there's a flush of activity that occurs, or a burst, and then it stabilizes into basal soil respiration. Traditionally, we've been interested in basal soil respiration. What is the steady state mineralization of carbon in the soil, which then also relates to the steady state mineralization of nitrogen, which is an element of prime importance in agriculture. So what we're trying to do is to standardize it by drying the soil first and then re-wetting it and create this flush. So this flush of activity is important because it actually relates very well to the basal soil respiration. It's not the same number, but it relates very well to it. So the three depths are really separated continuously throughout the incubation. And the incubation is relatively simple. It's a simple canning jar with soil in there, with an alkali trap that traps the carbon dioxide that's evolved from the soil into the alkali, because the alkali traps this carbon dioxide as a weak acid. And so it's trapped in there, and then we measure how much of that mineralization occurred during the first 3 days. So I've had a number of collaborations over the past decade or so, and these are some of the collaborations that I wanted to point out very intentionally, that the Virginia Department of Conservation and Recreation houses the Soil and Water Management Division. Soil and Water Conservation Division. And that's been an important partner in Virginia through the North Carolina Cooperative Extension throughout North Carolina has been very helpful in my studies, and the Cooperative Extension in South Carolina at Clemson University. All three of these states have strong extension activities and partnerships with farmers, and I've been really fortunate to work with them. And there have been some other ones too, like the North Carolina Foundation for Soil and Water Conservation. We've had several projects, demonstration projects that turned into research, and we've been able to demonstrate the value of conservation approach to soil health, to agricultural productivity, and to profitability. And then the Natural Resource Conservation Service in all three of these states, as well as surrounding states, has been very helpful. And again, I wanted to mention Cotton Incorporated, who has been dedicated in support of my research over the past 10 years. And more recently, the North Carolina Council, Forage and Grassland Council, that has sponsored some of the research and has been a network of activities, of farmer activities for me. And importantly, there are farmers involved in this. And these are two of the most common collaborators I've had in North Carolina, Mr. Zeb Winslow and Mr. Mike Jones, been very strong collaborators with me. And there are many, many others, and I'm going to show you a few other faces in the presentation. But these, having these strong partnerships with farmers has been really important for the research that I conduct. So I want to get into some numbers, and I hope that you'll bear with me with some of the numbers that they're important for me to illustrate the points that we want to make. Because in the science that we conduct, we need numbers to be able to tell you what the effect of the management is having on the soil resource. So soil test biological activity is measured in this milligrams of carbon dioxide, carbon equivalent of per kilogram of soil in 3 days. If we look at these this biological activity under conventional till with no cover, we see that there's greater biological activity near the surface in the top 4 inches, and then it declines somewhat exponentially with depth. And so basically the 4 to 8 inch layer is about half of that of the 0 to 4, and then the 8 to 12 almost half of that of the 4 to 8. So it's declining with depth. This is a natural consequence. This is how soils are in nature, that they develop with resources near the surface and they decline with depth. Now, if we looking at some of the farms that had no till and no cover, we see that the top 4 inches is enriched in biological activity compared to the conventional till cover. This is important aspect, and this shows that no tillage is having a protective value for the soil, and it's also enriching the biological activity. Now, it's not just important just to enrich the biological activity just for that sake, but because it actually gives us as land managers something that we can actually benefit from. And I'm hoping to illustrate that with some other data slides. But here also, it shows that the biological activity declines with depth. This is a natural consequence. It's going to be really hard for us to change biological activity deeper in the soil profile. It may be possible under certain circumstances, but there's always going to be this progression downward. But it shows here that there was an increase with no tillage at all 3 depths. But we see that by adding single species cover or multi-species cover, there's a progressive increase near the surface, but then less of an increase with depth. And so we were basically forming a more enriched cover of carbon resources near the surface, and that leads to this enrichment in biological activity. And this biological activity is now an indicator of the health condition of the soil. And here are some data that might just, yes, they just appeared on your screen. But this was based on a number of field trials. What we did was we looked at, we tested biological activity on the horizontal axis and associated that with grain yield response of corn to the initial dose of nitrogen fertilizer. So what happens when we apply, say, 10 pounds of nitrogen? Do we get some increase? Well, in this case here, these units are pounds of grain per pound of nitrogen. This graphic here is to illustrate that if we have low biological activity, we need to apply nitrogen fertilizer. Otherwise, there won't be grain production. So this means that with, say, 10 pounds of nitrogen, you would increase the pounds of grain up to 1,500. So 150 times 10. So it'd be 1,500, and so that would be an increase of, let's see, that'd be 3 bushels an acre, just from 10 pounds of nitrogen. So it's a pretty dramatic increase that occurs. But the point being that if we had high biological activity, we wouldn't need much nitrogen fertilizer. The soil is able to supply the nitrogen because that biological activity is actually activated, and it's recycling the nitrogen in the soil. And it can only have high biological activity if it had the resources there. So this is a little bit, maybe a more simple way of presenting this because if we apply nitrogen, typically we see an increase in grain yield and then a plateau. And that's what occurred in this study here. This is one example out of the 40-some trials that were conducted. But I wanted to point out that that initial dose is that first, tangential slope there that occurs. So in this case here, there would be 70 pounds of grain produced per pound of nitrogen. So that's a very high rate, and that would be something typical or something that we might expect or that we're told to expect. But this doesn't always occur. And it basically doesn't occur, and I'm just really summarizing because I don't want the focus only to be on nitrogen in this case here, but I wanted to wrap it up to say that this grain yield response to initial dose of nitrogen fertilizer can be related to this biological activity. And so this is from a number of trials. That's why there are error bars. It's based on the number of trials there. And basically, we can start putting this information into a recommendation system where if we had low biological activity, or in this case, very low, VL, we would need a high amount of nitrogen fertilizer. And that nitrogen fertilizer could be, would be scaled to the expected yield. And then if we had higher biological activity, we would need less nitrogen fertilizer. And more biological activity, even less nitrogen. And the more biological activity, the less nitrogen that we would need. And it follows this nonlinear description. And so that this is just telling us that there won't probably be a point where you can have no nitrogen fertilizer and get the optimal yield that you're looking for, or economically optimal. But if you did apply no nitrogen fertilizer and you had really high biological activity, you would get a very high yield. There's no doubt about it, as long as water is available. All right, so the practical approach to this prediction of nitrogen for corn grain is that we make some assumptions, and we had a number of field trials, but basically the average condition or the typical situation is that we apply 1 pound of nitrogen per bushel of grain for corn production. This is a value that most farmers understand because corn is grown in all 50 states of the United States and all around the world. And so you know that you need nitrogen to produce corn, right? That's the idea. And so the simplification of a USDA colleague's generations before me simplified this into like 1 pound of nitrogen per bushel of grain. Well, some use 1.2, some use 0.9. So that's why I've got a little bit of a range there. But that's just considering the yield goal. Doesn't tell us anything about what the soil condition is. And in fact, if we knew the soil condition, we actually would need to apply less nitrogen fertilizer. But the first point really is to make is that this nitrogen that's recommended as a typical is actually what we observe in soils that are deplete of biological activity. We need to have nitrogen and that's why this line crosses into this zone here. And in fact, it might go a little bit higher here. So maybe there are some sites that need a little bit more nitrogen. And you as farmers, you might understand that some fields just need a little bit more. But it's also possible that if you use a conservation approach, that you're building a biological activity, you could actually start using less nitrogen fertilizer. And nitrogen fertilizer is no longer cheap, and it is an environmental threat. So we need to pay attention to it. So improving soil health or biological activity could reduce the need for nitrogen fertilizers and protect environmental quality. I wanted to bring your attention now to a different phase of the research that I was conducting, and that really led me into the program with Dr. Lal at Ohio State and this quantification of carbon accumulation in soil. So we had an opportunity to sample the 25 research locations in North Carolina. We officially have 18, but there were some outlying stations associated with the main ones. And so there were 25 locations. And I sampled them across what we would call the Blue Ridge region in western North Carolina, the Piedmont in the center where it's hilly, and we have altisols, and then, well, all of them have altisols, and then the coastal plain and flatwoods where it's flatter, and we have typically more sandier soils, coarser textured material. All right, so I'm going to explain a little bit about the details that we got here. The association of biological activity with soil aggregation. So these are some aggregates in the background here, and we see that soil test biological activity has some association with mean weight diameter. So this is an estimation of aggregation. How stable are these aggregates in water? That's what we're trying to measure in that indicator there. And with conventional tillage, there's a slight increase in mean weight diameter with higher biological activity. So that's a good thing. We can actually till the soil, and if we incorporate enough organic material, then maybe it can lead to higher stability of the aggregates. But once we introduce conservation, when we disturb the soil less, when you introduce no-till cropland, we see that the stability of the aggregates is greatly improved. We see more of a water-stable aggregates with higher biological activity, and we can increase the biological activity and the stability of the soil if we have perennials in there, with grasslands in this case, or with woodlands in another case. So we see that there's actually an association of biological activity not only with nitrogen mineralization, but also with the stability of aggregates. And so we might have a common function. And basically it's showing that there's probably a point where if we increase the biological activity beyond 800 or 1,000, which I believe is kind of like the target where we should be at. I'm not sure that we need to focus on biological activity any greater than that, because it'll be really hard to maintain that level over time. But it basically is showing that okay, the stability of the aggregates is really starting to plateau at those high levels also. And there's an association that enriching your soil is also going to lead to less erosion. So in this study with the 4 different physiographic regions, we had 4 land uses. We had 100 fields total in each of the, essentially 100 fields total in each of the different land uses. And this diagram here, if we actually carved out the soil profile, we would see that soil organic matter is on the horizontal axis, and we're going deep into the soil profile, down to 24 inches. So what happens, again, this is similar to the one graphic I showed you on biological activity, but that organic matter is enriched near the surface, and then it declines with depth. This is a natural thing that is in the southeastern US and likely all over the world, although I don't have data from all over the world. But basically, we know about these profile data all around the world, and most soils react this way. The numbers might be different, of course, because of the climatic conditions. But basically, this graphic here is showing that there's concentration near the surface and decline with depth. Now, if we take the top 6 inches, which is a standard soil sampling depth, we would say that the conventional till cropland in this case and across North Carolina of the Piedmont would be 1.7%. Not necessarily an extremely low value, but a value that is lower than with no-till cropland and with grassland and with woodland. So we can increase the organic matter up to nearly 5% in the Piedmont if we had woodland management. So this is something that is not often thought about in our region, that we can actually have organic matter levels this high. But these were on numerous fields throughout the state. So with the biological activity, I wanted to express here this newer concept about root zone enrichment, because this is something that I believe that farmers will be interested in because of the carbon credit programs that are out there, and the need for having a degraded situation so that you can get paid to improve it. When in fact, hopefully we can have actually a system where we can make incentives and keep farmers that are doing good things to keep them doing good things. And this is a way that we can maybe start doing that. So that's the background of why I'm looking at this. So if we look at this soil test biological activity, again, it's high at the surface, it declines with depth, it follows a mathematical formula. And there are important things of why the mathematical formula, what it tells us. The 47 relates to that minimal level deep in the profile. The 957 relates to the upper level of how we can do that. So again, we need numbers to be able to quantify. And that's otherwise I would just be waving my hands, which I tend to do as well. But the numbers really quantify it. And then this coefficient then tells us how steep that curve is going to bend. Okay, but the important thing here is that nature provided something for all of the landowners. You didn't start with 0. you started with something. You bought your land, or you were inherited your land, and it started with something. And that baseline condition is what I'm really trying to find out. What is the baseline condition versus what you've managed over the past half a century or so? And so that's what this diagram is really illustrating. Of course, there are some numbers in there which I'm not going to go through. The important part is that red component, that red rectangle, is telling us that that's what was there. Deeper in the profile, we're not really able to change that dramatically. Now, there may be some slight variations, but generally, I would say that the assumption is we cannot really change that level deep in the profile. And that is what soil processes over millennia have provided to us. But what happens near the surface is what you as farmers are controlling. The management control. So that could be through land use, or it could be the type of management practice that you have. And that's where we need to focus. The red part is something that we need to quantify so that we know what is your starting point. And it would be nice to have measurements of the profile 100 years ago from all of the fields across America, but we don't have that. So we have to have other ways of characterizing that. I think that the red part is the red triangle, that rectangle there is actually telling us what is there, what could have been there 100 years ago. It may or may not be, but we have to have some starting point. But the green part is where we want to quantify. And that really relates to this graphic that I showed with you before, that soil organic matter is high near the surface, and then all those land uses, they converge to one point deeper in the profile. That convergence tells us something. And so the difference makers are really about land use. Broad land use characterization is important. And if you're cropping, using a conservation approach is going to be really important, disturbing the soil less, or if you're disturbing, getting to plants to regrow as soon as possible with cover crops or with animal manures. Those are important aspects. And there are a lot of different possibilities. And this project with the sea farm is really what I'm trying to characterize, a lot of different processes. And that's why I'm going to different regions. And then in the grazing, the managed livestock field, how we manage the pastures is going to be really important. And we're getting some useful data on that as well. Here I wanted to just characterize here some results that from the research stations that across North Carolina. This is the root zone enrichment of total soil nitrogen in pounds of nitrogen per acre. This is what management led to, and I've already subtracted off the petagenically controlled. There was significant amount of petagenically controlled. So I don't really remember the number, but I'm going to guess that the petagenically controlled was at least 300 pounds of nitrogen. And I think it was more than that. But the point here is that in the flatwoods along the coast of North Carolina, we saw an increase from conventional till cropland to no-till cropland to grassland, and then a slight decrease with woodland. So this is with total nitrogen. So grasslands typically have received nitrogen in the past, and there are some producers that are using less nitrogen these days. And so nitrogen is accumulating based on the management. There's an increase with conservation. In the coastal plain, essentially a very similar pattern is occurring. In the Piedmont, another similar pattern that's occurring. And in the Blue Ridge in the mountains, another pattern that is consistent and generally consistent. But with the point that I wanted to make is that the regions, we do need to characterize this across or among different regions, because there are unique soil characteristics that are important, as well as the environmental conditions. So in this case, the Blue Ridge has a little bit cooler climate than it does in the flatwoods or coastal plain. And so we see that's why there could have been a reason why there's greater nitrogen accumulation due to management under the grassland. And then overall, the main point is that conservation approach, switching from conventional to no-till cropland, is going to lead to an accumulation of nitrogen that benefits you now and in the future. If you deplete all of your nitrogen by destroying the organic matter, then you have to apply fertilizers. That's your only choice if you want to produce. So that this is creating a buffer. Conservation is a way of building resilience for the future. And grasslands, perennial grasslands, have a great ability to increase our nitrogen even more. And because woodlands are not fertilized, they probably have a nitrogen limitation, and that's why they're probably a little bit lower there. Same results, same trends, not the same numbers, but the same trends occurred for biological activity. And basically, again, this gradient with the environmental gradient is showing up very strongly as well. And again, biological activity is able to predict this nitrogen accumulation quite effectively. And so it's something that we can see that the soil is enriched in its activity. It's leading to this accumulation of nitrogen. All right, I wanted to just kind of let you know about some other projects that are part of the Sea Farm project. I've been tasked, I've taken the opportunity to explore different areas of the southeastern U.S. I have a home in Georgia. I work in North Carolina. I feel that the southeastern region is now my home. I've lived in the southeast U.S. longer than I have lived in any other place, and also where I was born in the central part of the country. So across Georgia, South Carolina, North Carolina, we have two land resource areas, the Southern Coastal Plain and the Atlantic Coast Flatwoods. And basically, I was targeting farms that had a conservation management. But although I was looking for opportunities, fields on these farms that had a diversity, I want to be able to characterize this root zone enrichment of carbon, of nitrogen, of biological activity, and a plethora of soil health attributes on these conservation-oriented farms, as well as some that are maybe more traditional, to get an idea, well, what's happening in the country. And so this is being done in the Southeast, in these three states. And these are some of the faces of the collaborators that I've been fortunate enough to meet and to sample on their farms. So I really appreciate their collaboration and their interest in the project. I've been on other sampling trips too. The Kentucky is perhaps a little bit outside of the southeastern U.S., but I've had an opportunity to work with Dr. Greg Halleck and sample some farms there, and we found some really interesting results that I hope to share with the farmers there really soon. I've just been able to finalize some of those data there. And I've recently been throughout Texas because Cotton Incorporated is funding my project. They wanted to see me in the state of Texas, where a lot of cotton is grown, and I was happy to be there, although I was not happy with the dust storms that I had to endure this past winter. All right, so the kind of the summary from this part of the presentation is really about you were given something, and what you were given is something that it may in the future be tied to, say, land value, and is tied to land value in some intrinsic way. But that starting point should not leave you at a low level. An opportunity exists for increasing biological activity near the surface, whether you were starting low point or a high point, but we need to characterize what the starting point was, because if we take the example from the fertile soils where my mother lives in eastern Nebraska, it's not fair to compare it with a soil in Orangeburg County, South Carolina, because the climatic and the soil conditions were different. They were just different. So we shouldn't be, say, having a carbon credit trading scheme based on the inherent conditions, but or the actual carbon in there. But what was the inherent condition, so that a soil with low level can actually have an increase, and that would be a good thing. So whether it's inherently low or high, it's the conservation management that really is going to improve the buzz in the soil. So I thank the sponsors from this Sea Farm project, and especially Cotton Incorporated. And I'm going to just take 1 minute to describe, well, a couple minutes, I'm sorry, but to describe some documents that I wanted to share with you, although I'm running a little bit short on time. I'm not going to go much longer, but I wanted, I'm going to give you a link where I think that you'll be able to find some of this information after the webinar here. So this is the biological activity as a function of sand. Sand is an important attribute in that it actually doesn't allow the organic material to bind to the soil particles. And so we have lower biological activity typically in the sand, although there's a large scatter there, and that's due to the management. So farmers have a great role to play as to how that relationship will occur. And I'm going to skip over this, but basically, I wanted to show that the biological activity is related to organic carbon in a general sense. There's a lot of scatter in this upper graphic. In the density of the soil, when we have high biological activity, we get low density, and that's good because that means there's more pore space, more opportunities for roots to grow, for the organic matter to reside, and it leads to better soil tilth. And then finally, the water stable aggregation, again, is related to biological activity, as well as biological activity related to nitrogen mineralization. So that document is going to be posted. I'm going to share that with you. And then I wanted to bring your attention also to some activities I've had in the last couple of years. Writing for hay and forage grower. If you're not aware, that if you go to this link, which is you can search for soils, I believe, and then it'll take you to this page where soils articles are. And I have a series of articles on called Sunrise on Soil, and this is where many of the articles, one-page articles that you might find there. And then I've also written for the Carolina Cattle Connection, a group of cattlemen's in the North Carolina, South Carolina on these topics. I'm planning to post this with Asmita on the CFARM website as well. It might be useful to you, and that's going to be located here at the website, the Carbon Farming Alliance for Research and Management. And then if you got down on the left side there, you might find them in the publications or presentations. So with that, I wanted to end and give you an opportunity then to have some questions if you so desire.
Awesome. Thank you, Dr. Franz Lubis. This is excellent presentation and data rich. I can see the whole almost 30 to 40% area of a US covered through this soil sampling data. This is awesome. I just want to let participants know Dr. Virginia Ridge couldn't join us today because of some medical emergency. We will definitely invite her again to talk about farming microbes in future webinar. And please feel free to ask if you have any question through question chat button or like a question answer section bottom of the Zoom, or you can raise your hand. I will unmute you and you can ask the question. Anyone? So Dr. Franz Lubers, just I can go for this. Now you have like really good data set and everything. And then, okay, we have one question here. I will ask my question later on. Prasanna, do you want to cover this question and answer session of the presentation?
Yes, sure. So the question is what's a reasonable depth you can control with management practices?
Okay, very good. Thanks for the question, John. Generally, what I'm targeting is the top 12 inches of soil. The top foot of soil is what we can control as land managers. Now, the roots, of course, penetrate deeper than that. But when you have roots developing down there, you also have decomposition of the organic material. So it's really the focus is on organic matter as a concept. And so the top 12 inches is where accumulation could occur. It's going to be mostly focused in the top 6 inches, but the top 12 inches is I think a reasonable one. And also internationally, we focus on the top 30 centimeters or the top foot as a depth of influence. And so please don't get me wrong that roots do penetrate deeper than the top 12 inches, but they also decompose down there, and there are very few roots that are down there. So that's why the focus is really about the top 12 inches.
That was great. Feel free to ask if you guys have any questions, or you can directly send a message.
can go next. One thing, like as a modeler, I can see like a really huge data set and like really good sampling point. I know we are in farm project, we are using this data for a modeling purpose and everything. So I was just wondering, do you have any like a modeling articles or any work you have done using this data set to predict where we don't have a really good information?
Yeah, so part of the project, we have a collaborator that is going to do some modeling. I've received a manuscript. I have not reviewed the manuscript yet, so I can't really say how much information is in there. But that is the intent, is that we want to use the information that we get from private farms, real farms that are out there, using management that they use, and see if the modeling exercise changes compared to what we've been traditionally doing through research stations, where everything is generally controlled, and these are valuable projects where they have long-term research. There's no doubt about it. A lot of things are controlled, but there are also management schemes on farms that we don't explore, and so it's really important for us to understand that as well.
Thank you. I think we have next.
Are there any commercial labs offering STBA yet? We have used Solvita tests, but have not correlated that your measurements.
Yeah, the salt has biological activity. I started my career as a PhD student at Texas A&M in 1990, 91, I think 91. And I started using this approach. I was told that we should not dry soil and then re-wet it and look at mineralization. But in fact, George Stanford, when he did his nitrogen mineralization studies, the soils were always dried, and but he just discarded the first 2 weeks of data, knowing that it wasn't steady state. And it's true, they was not steady state. But so getting to the question, yes, Solvita is a simple approach, and they use 1 day. I choose to use 3 days because I want to get a reliable number that I feel confident with, and I've used it for more than 25 years now. And so there are other labs that are offering approaches like the Solvita. They may use an infrared gas analyzer, which Solvita doesn't use, but there are oftentimes a 1 day approach, and Cornell Soil Health Lab, they use 4 days. And so these are variations that are a little bit, they can be calibrated, but I would just say that the most of the data, published data, are on the 3 day that I've used, and it just other labs need to calibrate with that. So yes, I believe we have a standard. It's just that we need to get those other labs to calibrate to this standard.
Thanks. We have one more question on this. In question answer. Have you observed a particular cover crop combination that are better for enhancing biological activity?
Nice question. I could have an opinion, and I'll share my opinion, but I would say that a lot of different cover crops can be used. First of all, I think mixtures have real value, but like the really large mixtures with 7-way or 9-way or 13 species, though all those are you can try them out and see what works, and I think that gives you value. But then when you have more of them, oftentimes it's more costly. I think that generally we need to consider the functional groups. We need grass, we need legume, and we need a broadleaf, succulent, more succulent type broadleaf, and that or some stemmy material too. It depends on what your goals are. But those functional groups, like so it depends on where you're at. I mean, if we're in the Southeast, I would say that that rye as a cereal grain is really good. I like actually the value of triticale. The seed might be more expensive or not available, and so that's an issue. And then, of course, legume, hairy vetch does really well. It climbs on the cereal, so that you can get a high biomass if you allow it to grow along. And those cover crops feed the biology if you keep them on the soil surface and build it over time. So getting a good system where you're decomposing the material on the surface means that the nutrients are then percolating through the soil, and the biological activity is maintained because the carbon resources are there continuously. And so again, I've mentioned some species, but I think there are a large range of species. Certainly in the grazing world, crimson clover is a good one, to overseed warm season pastures. There are others that should be considered as well. And I think what we need to find is that producers and researchers find additional species that could theoretically be working.
Thank you. Yep. Thanks. Uh, Anyone last minute question? Can raise your hand if you wanna speak instead of typing it. Awesome. Yeah. I think thank you very much for joining us today. Thank you very much, Dr. Franz Luber, giving us excellent presentation. And this is our a first talk. So we will have a couple of more webinars coming. Second webinar is in November 21st. Correct me, Prasanna, if I'm wrong.
Yes.
And there will be a 2 to 3 webinar in spring season. So we'll keep that webinar series going on for next another year, and we'll definitely announce those through our network, and please register for the another webinar. And I'm just gonna share my screen for CCA credit we received, so you can scan that QR code and fill out your information for getting that credit. Can you see my screen?
Yes.
I'm just giving a minute to scan it, and then
Asmita, Prasanna, thank you for hosting the webinar. I appreciate being on.
Thank you, Professor. It was valuable insights. Thank you. Thank you all for joining, and please stay updated with the upcoming webinars. We will try to bring as many useful informations as we can throughout this series. Thank you.
yeah. And one last thing, I think Dr. Franz Luber will share this information, and I will definitely share it with our registered participants, the links and some documents mentioned through this webinar. And I was gonna say something, I forgot again. Yep. And yes, and then this webinar recordings will be available on Ag Crops Team's YouTube channel. You will also get that link through email. Thank you very much.